Welcome everyone. Today we'll discuss practice problems related to acid-base biochemistry, stabilizing interactions in biomolecules, and biochemical thermodynamics. These questions will help you in better understanding of the concepts related to these topics. So let's start. The first question is, it's a multiple choice question where they have asked you to choose the incorrect statement out of the following. So you need to read the options very carefully. The first option is the random coil state, which is the unfolded form of the protein, has high entropy. Seems true because if you look at the different structural conformations of the protein, we have primary, secondary, tertiary, and in some cases quaternary structure. Now the primary structure is the linear sequence of the amino acids, and this is the one which is in a disordered state, in a random coil state. Compared to the other two, the secondary is the one where you will see the protein configurations rich in alpha helix or beta sheet. The tertiary is the three-dimensional structure of the protein, which is responsible for the functionality of the protein. Remember, when we are moving from the primary to secondary to tertiary, the entropy of the protein is getting decreased. You all are aware with the term entropy. It's the measure of disorder or randomness. So the primary structure, the linear sequence of the amino acids, is the one which is having a high entropy because it has a tendency, this sequence of the amino acids, they can rearrange themselves in a different combinations. You all are aware with the term Leventhal paradox, where these number of amino acids, be it like 10, 20, or 30, or even more than that, they have a freedom to reorient themselves, or they have the freedom to organize themselves in a different conformation. So the random coil state of protein has a high entropy because of the high degree of freedom. So this is true. How about the second statement? In an aqueous solution, when an enzyme binds substrate, there is considerable loss of entropy for water molecules. Seems false because when there is an enzyme and then you have something called, so let me draw the structure of some substrate. So before binding, what usually happens in case of in a solution form, be it inside the body or when you're performing the experiment in the lab, these enzymes and substrate initially, when they're not coming together, they are surrounded by the water molecules. They are either forming a hydration cell. These water molecules are either combining with the enzyme by forming a hydration cell, or these are showing some kind of electrostatic interactions with the enzyme molecule. Remember, majority of the times the enzymes are made up of amino acids, so the water molecules are going to interact with those amino acids. Same for the substrate. The water molecules will create a hydration cell. They are going to interact depending upon the nature of the substrate. Remember, water being polar, so it will look for the polar components in the substrate molecule. So this holds true for any of the moieties. Now once the enzyme and substrate will come into the close proximity, what is going to happen? These water molecules will be freed. They will move from these places. The point which I'm trying to make here is that these hydration cells will get broken down and these enzyme and substrate will form a complex. And by doing so, what is happening? The number of water molecules which were surrounding initially the enzyme and substrate individually, they are going to be less in number because some of these will be free. These water molecules will be free. And when I'm saying the water molecules will be free, what do you think the entropy is going to be? Entropy is the measure of disorder or randomness. So the entropy of water molecule will increase. Remember, I have to choose the incorrect statement. So this seems to be false. Third, increase in the entropy of the solvent plays an important role in the hydrophobic effect. Seems true here, because remember, when we talk about the hydrophobic effect, we talk about the apolar molecules or the non-polar molecules coming together. So let's say if I'm talking about the protein, the hydrophobic amino acids will start interacting with each other. They will form a hydrophobic collapse-like structure, from where you will find majority of the non-polar amino acids are getting buried at that point. And remember, as it is very clear from the name, if I'm saying hydrophobic, those are going to be water hitting. So those moieties are not going to interact with the water molecules. So they are generally buried inside. So let's consider you have the individual lipid molecules where you will see, the clathret-like structure of water molecules surrounding these lipid molecules. So you have numerous H2O surrounding these hydrophobic tails. Same for the phosphate moieties, but since we are talking about the hydrophobic, we'll not consider the phosphate ones. Remember, if I'm talking about a simple lipid, it's going to be a phospholipid, which is going to have a phosphate group as well as the two hydrophobic side chains. Same here, water molecules. Now, what do you think when these lipid molecules will combine? Same like this example, when these lipid molecules will combine, the number of water molecules which were surrounding these lipid molecules individually will now decrease. Now, when I'm saying decrease, the water molecules are now free. There will be increase in the entropy. So seems true. Fourth, for a protein to fold, the loss of entropy must be balanced by decrease in enthalpy by forming strong non-covalent interactions. Seems true. Remember the same thing which I was explaining in the case 1. If I'm moving from 1 to 2 to 3, there is an increase in the entropy of the water molecules. But what is happening for the protein molecule? Now the entropy of the protein by itself is decreasing. So there is a lot of biology happens when the protein gets folded. I hope you all are familiar with the term funnel theory of the protein folding. So the entropy of the protein by themselves is getting reduced, is getting decreased. But remember, this decrease, if you look at the famous Gibbs free energy equation, delta G equal to delta H minus T delta S, if then there is a decrease in the entropy, I need to balance this equation to make sure that the protein folding the protein folding happens. So for that, this decrease in the entropy or the loss of the entropy is generally balanced by the decrease in the enthalpy, which I'm talking about, this delta H parameter, by forming strong non-covalent interactions, which could be dipole induced dipole, London dispersion forces, cation pi interactions, what is salt breeze interactions, which is a combination of electrostatic interactions as well as the hydrogen bonding. So coming back to this question, what is the incorrect statement is going to be? It's the second one. Next question. Which one of the following regarding the thermodynamics of ATP, which is a adenosine triphosphate hydrolysis, is not true? Let me draw the structure of the adenosine 5' triphosphate first. Remember, we are going to have sugar. It's a ribose sugar. Then we are going to have adenine as a nitrogenous base. Remember, adenine has a purine ring. And then I need to draw 3 phosphates. Remember, these are termed as alpha, beta, and gamma. These bonds are called as phosphoanhydride bonds. What is now try to look at these options. So the first, which one of the following? So what I have to choose? I have to choose not true. That means incorrect. There is a less charge repulsion in case of ATP compared to ADP molecule. Seems false, because the ATP is the adenosine triphosphate. Remember, ADP is the adenosine diphosphate. There is one less phosphate molecule. Now, simple. We just count the negative charges present here. We have total 1, 2, 3, 4. There is going to be comparatively more repulsion. Now, in case of the ADP, what is going to happen? We are left with only 2 phosphate, adenosine diphosphate. So you are not going to see all the 3. You are going to have only the 2. So of course, if these 2 oxygens, if there will be a decrease in the number of phosphate molecules, there is going to be a decrease in the charge, and hence there is going to be a less charge repulsion. So less charge repulsion in case of ATP compared to ADP, false. The less charge repulsion is going to be in ADP and not ATP. Coming back to the second, the high energy of ATP molecule is because of 2 phosphoanhydride bonds. True. These are the 2 phosphoanhydride bonds which I discussed before, and they are having a high energy, and this makes this reaction spontaneous, which allows the ATP to hydrolyze into adenosine diphosphate and inorganic phosphate. Where you will find a break, this phosphate molecule will release, and ADP further gets broken down into AMP. So the high energy of ATP molecule is because of 2 phosphoanhydride bonds. Seems true. Resonance stabilization of ADP is greater than ATP. Seems true. Why so? Because in case of the ADP, again, like, you just need to compare this structure, which is ATP, with the ADP, which is going to be the same except this terminal phosphate. So resonance stabilization of ADP indeed is greater. Why? Because remember here, the lone pair of electrons which are present there on the oxygen is surrounded by a 2 electron withdrawing groups. I mean, you have this oxygen getting surrounded by a 2 electron withdrawing groups here, same here. Now this results in the, I mean, they are going to generate the resonance structure, but those are not going to be stabilized because of the presence of these 2 electron withdrawing groups. Now this is not going to be the case with the ADP, because this will not be there, and then you are only left with one of the phosphoanhydride bonds. Comparatively, the resonance stabilization is going to be more. I hope you are familiar with the term resonance, where there is going to be a delocalization of the electrons, resulting in the generation of structures. Fourth, ATP is an unstable molecule which hydrolyzes to ADP and inorganic phosphate. This was the same statement which I made it before. Yes, because of the presence of these 2 phosphoanhydride bonds, highly unstable, hydrolyzed to the adenosine diphosphate, which is comparatively more stable than the ATP and inorganic phosphate. So which one of the following regarding the thermodynamics of ATP is incorrect? And the answer is going to be 1. Next question. Again, it's an MCQ, and if you'll see, they have asked you which one of the following statements is true. So here I have to mention the statement which is true. The enthalpy of fusion of water is higher than its enthalpy of vaporization. Now remember, fusion is changing of state from solid to liquid. Vaporization is from liquid to gas. Remember, the value of both the enthalpy as well as the entropy of fusion of water is lesser than the enthalpy of vaporization, or even the entropy of the vaporization. So the first one seems false, because what I'm trying to say, when you move from liquid to gas, both the enthalpy as well as the entropy is going to be much higher than the enthalpy or the entropy of fusion. So this seems to be false. the probability of occurrence of a particular state depends upon the number of ways, which is also called as microstates, in which that arrangement can be achieved. So what it is said here is that the probability of occurrence of a particular state depends upon the number of microstates in which that arrangement can be achieved. Seems true. I'm going to explain it. Now consider a situation you have three quants and you are flipping those three quants. So there are several combinations which you can think of, like getting all the three head, getting all the three tail. But what I'm trying to say is that the probability, I'm not talking about all the combinations, I'm rather saying the probability of occurrence of a particular state. You will see, if you'll try to calculate the percentages, you will find there is a higher probability of getting two head and one tail, or one tail and two head, rather than getting these organized structures. So what I'm trying to say, you will see these kind of a combinations more compared to these organized ones, which are head, 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 or tail, tail, tail. Why so? Because just do some calculations and you will see the probability for these ones are going to be much higher than the three quants having the organized structure. So what I'm saying, the probability of occurrence of a particular state. So let's focus more on this state. So the two head and one tail. So the probability of occurrence of this state depends upon the number of ways in which that arrangement can be achieved. True. If I have to say so, that what is the probability of getting two heads and one tails, that depends upon the number of ways that can be arranged. So let's see, let's consider we have three quants. What are the different possibilities or the microstates? It could be either head, head, tail. It could be either head, tail, head, or tail, head, head. What else? So these are the different combinations for occurring this situation. If I'm going to compare this with this, there is only what is the probability of occurring of this particular state. The first is going to be head is 1/2 into the second quant having head is again 1/2, and the third having head again is going to be 1/2. So that is going to be 1/8. I hope I have not missed any other combinations. If so, you can just try to calculate it and you will find the percentage or the probability of occurrence of this state is much more compared to this one. Why? Because the frequency is much more, or the number of microstates for this combination is much more than having all the three heads or all the three tails. So it seems true. Third one is a spontaneous process tends to flow from high entropy to low entropy. So if you all can recall the second law of thermodynamics, for a spontaneous process to happen, the change in the entropy of the universe must be positive. So that should be from low to high. So the spontaneous process tends to flow from low to high and not from high to low. So this is again going to be false. The probability of organized state occurring increases with the number of molecules. Again, seems true. Let's discuss this part once again. So what I'm saying, read the last option very carefully. What I'm saying, the probability of organized state occurring increases with the number of molecules. So let's say we'll again consider the flipping of coin scenario. So we have two coins and I'm just considering the case of the organized structure. So what is the probability of getting head on both the two coins? And I'll keep on increasing the number of coins. 3. So two coins having the organized structure, 3 coins having the organized structure, and let's keep it 4 coins to 1, 2, 3, 4 coins having the organized structure. What is the probability of occurring this one? 1/2 into 1/2. How about this one? 1/2. And how about this one? 1/2 raised to the power 4. Now The probability of organized state, as you all can see, is decreasing and not increasing with the increase in the number of molecules. With the number of molecules, if I'll keep on increasing, the probability of organized state decreases. And how I can say an alternative statement about this statement, the probability of disorganized state occurring increases with the number of molecules. So again, which one of the following statements is true? The correct answer is option 2. Next question. Simple question. It's a very simple question. Out of the given following amino acids, which one of them has polarizable or easily polarizable side chain? Link is given for the structure, but it's always good to know the structure. Leucine, valine, isoleucine, phenylalanine, and glycine. So I'm asking out of all 1, 2, 3, 4, 5, which one of them has polarizable side chain? So if I have to draw glycine, it's going to be, correct me if I'm drawing a wrong structure. So this is glycine. How about the valine? This is valine. How about phenylalanine? So this is the one which I'm drawing right now is going to be the structure of phenylalanine. Remember, it has an aromatic. This is an aromatic amino acid because of the presence of this benzyl group. Phenylalanine in a single letter code is also represented by F. Valine as V, glycine as G. What are the other structures I'm left with? Glycine and isoleucine and leucine. Simple. So leucine is again, all except the phenylalanine, all the other amino acids are having a aliphatic side chain, while this one is carrying an aromatic side chain. So this is for leucine. How about the isoleucine? It's similar to the leucine structure. Simple. If you all can look at these structures, it's simple thing. The one having a pi electrons, those are going to be easily polarizable out of all these structures, out of all these 5. So 1, 2, 3, 4, 5. Out of all these 5 structures, as you all can see, the phenylalanine is having a aromatic side chain, which is carrying a pi electrons, which are going to be easily polarizable, and hence the correct answer is going to be phenylalanine, because those are the electrons which are going to be easily delocalized, which is also represented by a single letter code of F. Next question is the predominant force or the bond or the interaction helping in maintaining the secondary structure of protein is very simple question. So the secondary structure of the protein, if you see the major conformations of the secondary structure of the protein, I'm talking about alpha helix and beta sheet. Remember, in case of the alpha helix, the hydrogen bonds are intramolecular. In case of the beta sheet, you are going to see beta strands. The beta sheet is composed of beta strands, and within this beta sheet, it could be either parallel or anti-parallel. Remember, all these structures are stabilized by hydrogen bonding. Remember, hydrogen bond is not a real bond, but this is how the researchers have named it. So all these structures are stabilized by the hydrogen bonding. The next question is, it's related to the acid-base biochemistry. Very important question. We'll solve it. Zinc can act as a Lewis acid and coordinate amino acids like cysteine by their sulfahydryl groups. Let me draw the structure of the cysteine. So this is the backbone. And then you have the side chain. Remember, the cysteine is a sulfur containing amino acids. So zinc can act as a Lewis acid and coordinate amino acids like cysteine by their sulfahydryl groups. Normally, the sulfhydryl group of cysteine has a pK given. pK of it, I hope you all are familiar with the pK. The easiest way to calculate pK is the negative log K. K is the acid dissociation constant. The stronger acids have higher K and thus lower pK. However, the interaction with zinc can lower the pK of sulfhydryl groups by as much as 4 pH units. So let me draw another structure, or you can think of another structure where the zinc is coordinated to this structure, lowering the pK by 4. So it's going to be, initially it was 8. It lowers this value by 4 pH units, so it's going to be 4. Using your knowledge of acid-base chemistry, estimate the approximate ratio of protonated to deprotonated cysteine at neutral pH. So what we need to calculate is the protonated form of a cysteine, the ratio of protonated form of the cysteine to the deprotonated form. So the protonated form is going to be the one where the sulfur is bound to the hydrogen, and the other one is going to be where there is no hydrogen. So simple. If I have to calculate the protonated to deprotonated form, I need to use the famous Henderson-Hasselbalch equation, which is pH equal to pKa plus log base by acid. Remember what they have asked? They have asked the ratio of protonated to deprotonated form. So at the end we'll reverse it, but first I need to calculate it. Free cysteine, what is that going to be? pK given 8 and pH. Is that pH missing? However, the interaction, zinc can act as a Lewis acid. Neutral pH, they have given the neutral pH, so you can consider 7. So as it's given in the question, neutral pH 7, 8, and then we can calculate the ratio. So 7 minus 8 minus 1, then you need to calculate, you need to take anti-log, and the value is going to be 0.1 equal to base by acid. Remember what I need to calculate for the A part is the free cysteine, which I am doing so, but I need to keep the ratio in what form? Protonated to deprotonated. So I need to inverse it. So the value is going to be 1 by 0.1 equal to acid, which is the protonated form, by base. This is going to be, either you can write 1 upon 0.1, or you can write as 10 by 1, because remember this is going to be the same. So for the first part, the answer is going to be acid by base. Remember, I need to keep the acid wall apart on the numerator. Why so? Because that is the way in which they have asked the ratio of protonated. So acid is the one which is going to be protonate from your basic acid-base biochemistry classes. So the A part is 10 by 1. How about the B part? What I need to do, I need to use the same formula. What should I consider for the pKa for the B part is 4. So 7 minus 4, 3, and then the anti-log of 3, which is going to be 1000 by 1 equal to same base by acid. Remember, now again I have to inverse it because they have asked in a form of acid by base. So the B part is going to be 1 by 1000. Simple. I hope this solution is clear to each and every one of you. If not, you can raise your query in the comment box. Next question. It is generally assumed that the completely uncharged form of a neutral amino acid shown below does not exist in solution. So if I can look at this amino acid structure, it's a glycine. Look at the R, the side chain has only hydrogen atom. Instead, the major species of, for example, glycine given at pH values around pi is the isoelectric form. The major species they are talking about is the one shown on the right. Assuming pK value given 2.5, 9.5, estimate the fraction of the total glycine present as the rare uncharged form at pH 5.5. So they want to know the fraction of the total glycine present as this form A. So let's give this as A, this name as BA, as B. Simple. So what I need to calculate, first I need to understand or look at the structure of glycine very carefully. So if you can draw the structure of a glycine at a very, very low pH. We are going to have this structure. If I'll go beyond the pK value, which is 2.5, what I will get is the CS2. If I'll keep on increasing the pH and we'll go beyond 9.5, remember this is carrying +1 charge. This is the one which is going to be neutral isoelectric species. And then the third one, if I'll keep on increasing the pH and we'll go beyond 9.5, what I'm left with is the H2N. Simple acid base fundi you need to apply. H2N, CH2 and CO minus. Here it's carrying -1 charge. Now, what I need to do, I need to calculate the concentration of the fraction of this species. I mean, to be very honest, this is very, very rare species, because if you all can see, the carboxyl group is in the protonated form, that should be fine. But at the same time, the H2N is in the deprotonated form. So indeed, it's a rare uncharged form. But anyways, since we need to calculate it, we'll do it for this question. So what we will do, we'll conceive again the same, because these are the different forms. I need to use the famous Henderson-Hasselbalch equation, which is pH equal to pK plus log base by acid. The two pK values are given. So the first one we'll do it for the 2.5. pH they have asked us to calculate 5.5. Plus log B by A. Remember, at pH 5.5, I need to consider, like, with this pK value, what is going to be the base one and what is going to be the acidic form. Now, if you'll see, if I'm considering a pH 5.5, I have moved beyond the pH 2.5. So ideally, the majority of the species should exist in this form, and then the higher percentage is going to be this isoelectric species. But since they have asked us to calculate the rare uncharged form, I need to use this formula. What I'm trying to say, in this particular case, I need to consider this as an acid and this as a base. Simple thing. Remember, the carboxyl group is protonated. Here, this is deprotonated, and then, so we'll name it A, B, and C. So what is the value going to be? Same, 5.5 minus 2.5. We are left with 3, then anti-log 3, which is going to be 1000. Base by acid. Now, I need to calculate it for another pKa value, which is 9.5. And for that, what I need to do, I need to consider pH 5.5, 9.5 plus log base upon acid. Now, what is that value going to be? That's going to be -4, and the shift log, which is, I'm using the scientific calculator. So the anti-log value is going to be 1 into 10 raised to the power -4. This base upon acid is going to be 1 into 10 raised to the power -4. Now remember, in the second case where we have taken 9.5 as a pKa, you just need to use your concept that in that particular case, which I'm talking about here, when I've crossed the, when I'm at pK value 9.5, this is going to work as an acid, and this is going to work as a base. Why so? Because remember, H2N group is protonated here, and this one is deprotonated. So I need to, so this isoelectric species is now working as an acid, which is in the denominator. Here, the isoelectric species is working as a base, which is there in the numerator. So I need to use the same format. So what I'll do, I will change one of them. So I hope you all are getting, because I'm dealing with the, I'm keeping one thing constant, which is the isoelectric species, because that is the one which is going to be present in the major fraction. So if I'll inverse this one, this is going to be 10 raised to the power -3 equals to acid upon base. Remember, in this particular case, the acid is isoelectric, the base is isoelectric species. Here, if I have to say so, the base is isoelectric species, which is in the denominator. Here, the isoelectric species is acid, which is in the denominator. So fine. Now coming back, assuming pK1 this one, pK2 this 9.5, estimate the fraction of the total glycine present as the rare uncharged form. What are the fraction which is going to be in this rare uncharged form at pH 5.5? And what I need to do, I just need to get this species, which is in the form of RCH NH2, RCH NH2 COH, and if I have to get the fraction of the total glycine present in this form, that is going to be a combination of 10 raised to the power -3 into 10 raised to the power -4, which is going to be equivalent to 10 raised to the power -7. Look at this number, very, very, very small. So this is the one which is, so this rare uncharged form is going to exist at this much concentration at a pH of 5.5. I hope this solution is clear to each and every one of you. This is the last question. an enzyme catalyzing the decarboxylation of lysine accepts only the isoelectric form as substrate. What is the actual concentration of isoelectric lysine in a 10 raised to the power minus 3 molar solution of lysine in a buffer at pH 7.6? First of all, it's always good to draw structures. So what is the structure of lysine going to be? Remember, when I'm drawing such kind of structures, and don't get confused with the structures which I drew it in the previous slide. This one is the one which is, I'm drawing it for the lysine, which is also denominated by letter K at a physiological pH. That means pH 7.4. Now, if I'll go beyond the first pK value, which is 2.18, I'm just starting from the pH 0. So let's start with the pH 0, where every functional group is going to be protonated. If I'll move beyond pH 2.18, what is going to happen? This is the one which is going to have 2.18 pK. So this carboxyl group will lose the proton, will become CO minus, and then we have NH3 plus. Here overall we are having 1 plus 2 charge, here plus 1. Next, if we go beyond 8.95, we are going to get S3N, not S3N, because this is the one which is going to have the pKa value 8.95. So the deprotonation will take place. So H2N, CH, CO minus, and then CS2, 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 and H3 plus. Remember, this is the one which is carrying 0 charge. It's an isoelectric species. This is the one they are talking about in the question. So an enzyme catalyzing the decarboxylation, removal of carbon dioxide of lysine accepts only the isoelectric form, this species, as a substrate. What is the actual concentration of isoelectric lysine? So they are talking about this species concentration in a 10 raised to the minus 3 solution of lysine in a buffer at pH 7.6. So when I'm talking about 7.6, and I'm interested in knowing the concentration of the protonated and deprotonated form, I need to use the Henderson-Hasselbalch equation, which is pH equal to pKa plus log base by acid. What is the pH they have given? They have given a buffer of a pH 7.6. And then I need to consider a pKa. Remember, these species, I mean, they are going to present, they're not going to present at a pKa value of 2.18. I mean, very, very less might be, because a similar kind of question was asked in the previous slide. So remember, if I'm talking about this isoelectric species, they are not going to be present at a pKa of 2.18. Same holds true if I'm comparing the pKa of 8.95. I mean, these species are going to be very, very less. The point which I'm trying to make here is that since I'm dealing with this isoelectric species, I'm interested in this isoelectric species, and they are going to form beyond 8.95. That's the reason I'm taking this as a pK as 8.95. I'm not interested in 2.18 because they will only form once I will cross the pH value of 8.95. And then log base by acid. Again, you need to be very clever. If I'm talking about these two reactions, remember, there is one more step ahead where we'll raise the pH beyond 10.53, and then the side chain amino group will get deprotonated, and overall we'll generate a structure carrying -1 charge. Since I'm not interested in that structure, I have stopped here only. Coming back, look at these structure A, B, C, and focus more here between the conversion between the B and C part, and tell me which one of them is the acid, which one of them is the base. Simple. In this particular reaction, the B is going to work as an acid because amine group is protonated, while this isoelectric species is going to work as a base. So log, I'm just writing isoelectric species to simplify the things, which is going to work as a base, and then you have acid. This is because they have asked the concentration of isoelectric lysine, so it's good to have this isoelectric species in the numerator part. That will make our calculations easier. So 7.6 minus 8.95, which is going to be minus 1.35, and then I need to take antilog, and the value which you all will get is going to be 0.0446 equal to, it's the ratio of isoelectric species, the concentration of isoelectric species by concentration of acid. Remember, I need to calculate the concentration and not the ratio. So what is the concentration going to be? Isoelectric species concentration is going to be, sorry, (0.0446) divided by, I need, because the ratio is 0.0446 by 1. So the concentration of isoelectric species, which is the lysine in a near neutral state, is going to be present in the form of 0.0446 divided by 1.0446 of total. What is the total value? So that is given there in the question paper. It's the, what is the actual concentration of isoelectric lysine in a 10 raised to the minus 3 molar solution of lysine. So 10 raised to the minus 3, and what is the answer going to be? So it's 0.0446, 0.0446. You all can use calculator to solve it. And you will get 4.26 into 10 raised to the minus 5 molar. So this is going to be the final answer. I'm talking about the concentration of this isoelectric species, which is carrying a overall 0 charge. I hope these solutions are clear to each and every one of you. If not, you can address your query in the comment box. I'll be happy to answer it. So feel free to comment and ask your query. Thanks.